Next up, we're going to add another microbe to our conversation. So I'm happy to introduce Charles Rabkin. He's a senior investigator in the Infections and Immunoepidemiology branch within the Division of Cancer Epidemiology and Genetics at the NCI. And he has done seminal work on HIV-related malignancies, but we are delighted that he's moved into gastric cancer as well, and he'll talk to us about EBV.
That's an introduction, and I want to thank Robin Juho for bringing me to this meeting and allowing me to help contribute to all the important science that's being shared here. I think I'm included in the program, and not to knock H. pylori's importance, but to get this group to think about something that's been recognized for a long time, but mostly overlooked, that a small fraction, about 8 to 10% of gastric cancers, have Epstein-Barr virus in the tumor cells, and that wasn't always considered to be an important observation. But there are a number of molecular features of Epstein-Barr virus positive gastric cancer that really warrant attention. It's present in every tumor cell. It's got a monoclonal terminal repeat sequence indicating that it is present at the time of malignant transformation. It expresses what's thought to be a viral oncogene, and there are a number of clinical features of EBV positive gastric cancer that differ from from EBV negative gastric cancer, suggesting it might have importance both in etiology and prognosis. One thing that isn't well appreciated is that because gastric cancer is such a common disease, we've seen what the burden is worldwide. The fact that the small percentage of gastric cancer is EBV positive makes it constitute the largest fraction of EBV-associated malignancies in the worldwide cancer burden. So it outnumbers these other diseases on the slide that are more commonly thought of as being the EBV-associated cancers. But the problem is it's a minority, just like we're interested in the problems of health problems of minorities. EBV positive gastric cancer only represents a small fraction of the disease overall, so it's difficult to study. Any particular study of gastric cancer will only turn up a small number of EBV positive gastric cancers. So about 15 years ago, my colleagues and I at the National Cancer Institute tried to solve that problem by developing a consortium of studies of gastric cancer, that none of which had enough power on their own to look at EBV positive gastric cancer. Pooling our resources, bringing together the specimens and the data, we've got a group of investigators that are able to pay attention to this small fraction of gastric cancer that adds up when you consider it as to a large number of people with disease. And so putting together the findings that have been made in individual reports, there are a number of features that can come to light just by doing a meta-analysis. And some of those features are that EBV is very prevalent. Some of these are old findings. It's very prevalent in this unique histopathology that looks like nasopharyngeal carcinoma called lymphoepithelioma-like carcinoma. It has 80 to 90% of those tumors are EBV positive as compared to the run-of-the-mill gastric cancer that has a much lower prevalence. The prevalence of EBV is fairly similar worldwide, although it does seem to be a little bit higher in the Americas and a little bit lower in Asia, where the highest burden of gastric cancer is found. But overall, if you average across high incidence and low incidence countries, it seems to be a similar fraction. So as gastric cancer incidence fluctuates from population to population, it's always the same fraction of gastric cancer that's EBV positive, suggesting that it somehow collaborates with the pathway rather than representing a different path that has different etiologic factors, because otherwise, if that were the case, you'd expect that in low incidence areas, there'd be a bigger fraction of EBV positive gastric cancer, and that's not what we see. It is much more frequent in males, which has brought forward some hormonal hypotheses that we've tried to investigate. And it differs in the anatomic location in the stomach where the gastric cancers occur. It's much more frequent in the proximal parts of the stomach, and where in the antral sites of disease where H. pylori is most intense in its colonization, and where the bulk of gastric cancer arises. It's a somewhat lower fraction, although it does occur in that location as well. One of the most interesting features in my mind is that EBV positivity is found very frequently in the regeneration tissues and in post-surgical remnants. People that have gastric resections, who used to have gastric resections back in the old days for acid control or gastric resections for gastric cancer. In both of those conditions, the gastric cancers that subsequently occur in the remnant tissue have a much higher prevalence of EBV positivity, which seems to me to be a clue that EBV has an important role in the malignant transformation that occurs in the milieu of healing and regeneration after surgical injury. So the resources of our international EBV gastric cancer consortium have allowed us to pool data across studies and make some observations that can't be made by individual studies and therefore aren't available to just do compilations of the literature. Pooling the data, we were able to show that EBV positivity is more frequent in younger gastric cancers that occur in young individuals. We saw again the predominance in male gastric cancer and the anatomic subsite localization. We also observed that it's much higher in diffuse type gastric cancer. So put your thinking caps on and see how that plays into what we know about the differences in the etiologic sequence to intestinal type and diffuse type gastric cancer. And it at least in the work that we did here, there was no relationship to the clinical stage. We're also able to look at risk factors, and one risk factor that does come up as being significantly associated with EBV positivity is cigarette smoking, which increases the risk, as everyone knows, of gastric cancer overall. It also increases the conditional probability of EBV positivity among people that do have gastric cancer. So if you add the effect of smoking and the effect of sex, male smokers have about a fourfold greater prevalence of EBV positivity in their gastric cancers than in other groups. Conversely, there's, despite the weaker association of alcohol consumption with gastric cancer risk, there's no association with EBV positivity. Here's where it gets interesting from the clinical perspective. EBV positivity has a ameliorative impact on the prognosis of gastric cancer, and by summarizing across our consortium, we found about a 30% improved survival of people with EBV positive gastric cancer compared to EBV negatives. Our study also involves using the biospecimens across these various collections, and we were able to look at circulating inflammation markers, which may be either effects or causes of gastric cancer, but this is post-diagnosis blood levels of different inflammation markers. We have a panel at the NIH that we've been applying to different types of cancer. And with this 92-plex of inflammation markers, we found that EBV positive gastric cancers had significantly higher levels of 8 of these 92 markers as compared to people with EBV negative gastric cancer. And you can see that list on the left there, each of them have about a 2 to 4-fold association with EBV positivity. We took those findings, Constanza Camargo had this very clever idea to compare the levels in the blood to what's expressed in tissues. And so accessing TCGA data, which is another group I work with, we looked at the tissue expression of those same proteins, and in each instance, the 8 markers that have higher blood levels in EBV positive gastric cancer were also overexpressed in EBV positive gastric tissue compared to EBV negative gastric tissue. So there's important correspondence. Again, the cause versus effect relationship is not something that this would reveal. one of the problems in studying EBV-positive gastric cancer is the fact that it's not commonly assessed in clinical practice, and it's not necessarily tested even in groups that are studying gastric cancer. So we've been searching for better ways of identifying EBV-positive gastric cancer, particularly with an eye towards using blood samples from gastric cancer studies or prospective studies in which people do develop gastric cancer. And one of the ideas is to look at the antibody profiles of people that have EBV-positive gastric cancer and see whether that has some specific markers. So this is a collaboration that we've established with Josh LeBaer at the Arizona State University, where his group has developed an immunoproteomic approach to expressing all sorts of antigens on chips that can then be displayed and used to mine the antibodies of people with different diseases. And here I'm showing you a volcano plot of 89 different open reading frames from EBV that have been assessed for comparing people with EBV-positive gastric cancer to EBV-negative gastric cancer. And the x-axis shows the fold change for EBV positivity compared to EBV negativity, and the y-axis is the significance level. And you can see that there's a number of proteins that are labeled that are markers of EBV positivity being higher in the EBV-positive gastric cancer. Interestingly, all of the IgA antibodies that were examined with this chip were very similar between people with EBV positive and EBV negative gastric cancer. I'll remind you, it probably goes without saying, everyone is infected with EBV, but we're looking for markers of the presence of EBV in the tumors. And so IgA is not differential. Anti-EBV IgA is not differential in EBV-positive gastric cancer, but there are a number of antibodies, IgG antibodies, that are higher in people with EBV-positive gastric cancer. That chip is not really suited for large-scale mining of large numbers of samples. So their strategy at Arizona State is to take the top markers and put them into ELISAs in order to be able to ramp up the testing and find a way to identify these more useful antibody differences. And here, those same proteins that I had labeled on the volcano plot here are results in ELISA testing shown as a heat map. And you can see on the left-hand side, the EBV-positive tumors have a large number of positives compared to the EBV-negative tumors on the right. So that you get an impression from this graph, that at least in this first set of samples that were also tested on the chip, that combining several markers might be a way of identifying EBV-positive gastric cancer patients just by doing blood tests. Obviously, we've heard this morning about overfitting problems, and so it's very important. One strategy to overcome that is to validate these findings and apply the same test to another set of samples, and that's what we've done here. And those same assays were applied to a completely independent set of samples, and the markers that were tested had individual associations between 2 and 100-fold with EBV positivity of the tumors in people with gastric cancer, with AUCs that are quite promising for use as a biomarker, ranging up to 0.85, and starting to do some of the statistical calculations and develop an algorithm, just a very simple approach of counting the number of antibodies. If you just ask for the presence of 3 or more of the 7 validated antibodies, that has a sensitivity of 71% for identifying EBV positivity with a specificity of 93%. So we're getting towards the ability to identify EBV positivity just by antibody testing. It's taking a step back and looking at the whole spectrum of antibodies that are generated in EBV-positive gastric cancer. It's telling that it's not the antibodies, it's not proteins all the way throughout the genome that are generating the differential signal. It's particularly the immediate early lytic and the early lytic proteins in the EBV replicative cycle that are differential in EBV positive gastric cancer compared to EBV negative gastric cancer. Now, whether that could be directly interpreted as what is the replication state of EBV in gastric cancer, or is it just merely a byproduct of the same pathways that are triggered in EBV positive gastric cancer that also trigger these proteins, that again is not something that this particular data set is going to be able to answer. I've taken my interest in EBV positive gastric cancer to other groups, and just like I've here trying to get everyone here to think about it, I've worked with TCGA to remind them about the importance of EBV in gastric cancer and try to mine their data and apply it to understanding the role of EBV in a subset of tumors. As everyone knows, TCGA is a multi-omic characterization of different types of tumors with different types of assays and platforms, looking all the way across the genome, and it's very useful for doing all of these kinds of cross comparisons among different combinations of those factors. And one thing that comes out from TCGA is that EBV positivity is not a common finding across the tumor spectrum that's been examined. The STAD is the abbreviation in TCGA for gastric cancer, and you can see that the high numbers of EBV sequences are really only present in gastric cancers compared to all of these other tumor types that are shown on that slide. So what can we learn from TCGA data? The one approach is to let the data speak to us. So here's unsupervised clustering, and this was some of the earliest findings from TCGA, and it really was beyond my wildest expectations that we'd have such a dramatic difference. If you look at the methylation profile of EBV, of gastric cancer patients and just let tumors and do unsupervised clustering analyses, there's one cluster that's so intensely methylated that it clusters together, and those tumors that are most highly methylated and just the unsupervised clustering puts together are all the EBV positives, and it excludes all the other EBV negative gastric cancers. So it's uniquely highly methylated in the CPG islands of the promoter regions. In fact, one of my TCGA colleagues calls EBV positive gastric cancer the king of methylation, and this is a slide that we put together sort of for fun. You can see the four molecular subtypes of gastric cancer that we ended up nominating. The EBV positive gastric cancer shown in red has by far the highest level of promoter methylation across all of the TCGA tumor spectrum. The other types of gastric cancer are heavily methylated, as many in this room appreciate, but they pale in comparison to EBV positive gastric cancer. And there are some other molecular features that go along with EBV positivity. It's very different from other groups of gastric cancer based on the mutation profile, the chromosomal rearrangements, specific presence of mutations, lack of P53, for instance. So there are a number of other molecular features that make EBV positive gastric cancer very unique. One thing as a molecular epidemiologist that I didn't anticipate is that EBV positive gastric cancer had a similar set of molecular features regardless of the underlying etiology of the gastric cancer. And I'm showing that in this slide, that the cardiac cancers, as everyone appreciates, where H. pylori is not an important factor, there's the same molecular features of EBV positive gastric cancer in the cardia, where we don't think H. pylori generates the tumors, as EBV positive gastric cancer in the rest of the stomach, where most of the cases that were analyzed in TCGA were H. pylori induced. So it's a curious conundrum, still trying to tease out how it is that EBV positivity contributes to these two different major etiologies of gastric cancer. We've taken those same molecular approaches and the molecular characteristics of gastric cancer, and this was a suspicion even prior to the analysis that the esophageal cancer would have a lot of similarity to gastric cancer, and we did find that the lower esophageal cancers have molecular features that are very similar to gastric cancer, but they're of a specific type, that the chromosomal instability type of gastric, molecular subtype of gastric cancer, those same features are present in esophageal tumors of the lower esophagus. But there is no EBV in the lower esophagus. So it's the same, but it's different. Lower esophageal cancer looks like gastric cancer, but it doesn't get EBV. Taking that same approach, collating all up and down the GI tract, looking at the molecular features, it gets harder and harder to, and it was really an unanticipated finding that the EBV positivity was the most distinguishing characteristic of the molecular features of gastrointestinal tract adenocarcinoma up and down the GI tract. So if you took, we took all the TCGA data for gastrointestinal tract adenocarcinoma and tried to sort out what were the most important features to be able to develop a classification system, EBV positivity fell out as the strongest feature. It's just in the stomach. So there's something very unique about EBV positive gastric cancer that makes it a fascinating thing that we're still trying to understand. We've taken the findings from the data from TCGA and looked at what is going on from the virus side, and as I mentioned, with the antibody profiles being restricted, it's the viral genes that are expressed in EBV positive gastric cancer are not found throughout the viral genome. It's just one particular portion of the viral genome that is highly expressed in EBV positive gastric cancer. The BMH1A region of the genome has very high levels both of messenger RNA and the microRNAs that are found in EBV positive gastric cancer. And in fact, the microRNA levels of EBV positive gastric cancer that are derived from virally encoded sequences seem to almost outnumber the host sequences, despite the fact that EBV is a very tiny fraction of the DNA content. And there are host differences as well in EBV positive gastric cancer, host gene expression differences. And one of the things that was apparent just from the analysis of extracted nucleic acids is that certain important pathways are overexpressed in EBV positive compared to EBV negative gastric cancer. And PD-L1 is a pathway that's been recognized as a potential target for clinical treatment. In fact, has high levels of expression in EBV positive gastric cancer. But the TCGA approach, just using extracted nucleic acid, you can't really determine where these signals come from. So we were able to take both TCGA data, TCGA tumor tissue and our gastric cancer consortium tumor tissue, and looked under their microscope for the tissue distribution of PD-L1 that is overexpressed for a number of different, by a number of different mechanisms, both mutation and amplifications as well as non-amplification bases for overexpression in tumor cells. Well, it's not just the tumor cells that have high levels of PD-L1 in people with EBV positive, in tumors with EBV positivity. It's found both in the tumor cells, as shown on the left, and it's shown by examining the distribution on histologically. It's found in the immune cells, and it's in a specific pattern of the immune cells. It's found in the tumor infiltrating cells, and not just the cells that are on the margins, invasive margins of the tumors. So it's and it's not clear whether the tumor positivity or the immune cell positivity, the stromal positivity, is the better signal for nominating a particular patient for treatment with treatment directed against PD-L1 as an immune checkpoint inhibitor therapy. But to just kind of recap what we're able to find with all of these studies, it looks like EBV positive gastric cancer has a distinct etiology. It's got a better prognosis, which it makes us excited about the clinical implications. As the molecular data suggests, that it's got a unique pathophysiology. And based on the PD-L1 profile, it's been move forward in immune checkpoint therapy trials. So pembrolizumab was FDA approved for gastric cancers that express PD-L1, and this very kind of pivotal study from Korea found that EBV positive tumors had a 100% overall response rate. A tiny study, 6 EBV positive cases in that study, but the immune checkpoint therapy looks very promising, and there are other trials that are ongoing to see whether EBV positivity would represent a good clinical marker for using immune checkpoint treatment. So our work's not done. I hope this group can help contribute to it, trying to identify additional risk factors. Unfortunately, despite the resources of our group, we still don't have enough biospecimens. So if people have leads on how to get more biospecimens from patients that have either known EBV positivity or access to paired tumors and blood samples, that would be a great boon to this work. We're honing in on a blood test so that we can identify EBV positivity even without testing tumors, and that will make this work go better. And it's hopefully going to have direct applications either on the prevention side as an additional motivation for prevention of EBV. There's a lot of work that's going on with EBV vaccines to prevent that infection and the elimination of 8 or 10% of gastric cancer by preventing EBV from ever arising. That would be another thing to go into the benefit calculation of a preventive vaccine for EBV. And more immediately, the use of antivirals and immunotherapies in gastric cancer treatment is really going to be assisted by the direction towards EBV. So I think there's a lot that would be worthwhile studying more in this area. I have to acknowledge that we have a large number of collaborators, both people in this room and people outside, that have made this work possible. And we're always trying to increase the interest in EBV and the numbers of investigators that work with us. And really appreciate people's attention.